The autonomic nervous system, or ANS, responds to unconsciously perceived visceral sensations. The control center for the ANS is primarily the hypothalamus, but parts of the spinal cord may also be involved. The output of ANS results in inhibition or excitation of effector organs, including smooth muscle, cardiac muscle, and glands and adipose tissue. The ANS is composed of two pathways, the sympathetic and the parasympathetic, both of which regulate the function of many effectors. The regulation of a single effector by both sympathetic and parasympathetic divisions is called a dual innervation. Dual innervation will balance an autonomic effector's function. The motor pathways of the autonomic nervous system differ from the somatic nervous system in several ways: the number of motor neurons for each pathway, the presence of ganglia, and the number of and type of neurotransmitters. In the somatic nervous system, a single motor neuron excites the effector, which is a skeletal muscle in this case. In the autonomic nervous system, the motor pathway consists of two motor neurons, one following the other, carrying an action potential to the effector. The first neuron is an autonomic pathway, the preganglionic neuron, which is myelinated neuron. The first neuron synapses with the cell body of a second, non-myelinated, postganglionic neuron at an autonomic ganglion. In autonomic ganglia, the neurotransmitter acetylcholine is released via exocytosis from the preganglionic neuron. Diffusion across the synaptic cleft generates action potential on the postganglionic neuron. The action potential then continues to travel along the axon of postganglionic neuron toward the effector. Ganglia of the parasympathetic divisions are close to or within the effector organs. Because the preganglionic motor neurons come from four cranial nerves and the sacral segment of the spinal cord, this division is called craniosacral division. In a parasympathetic ganglion, there are only a few postganglionic neurons. This pattern often provides stimulation to only one effector. Ganglia on the sympathetic ANS are near the spinal cord or major abdominal arteries. Since the preganglionic motor neurons come from the thoracic and lumbar segments of the spinal cord, this division is also called thoracolumbar division. In a sympathetic ganglion, there are many postganglionic neurons. The pattern often provides stimulation to many effectors. In motor pathways, the way autonomic neurons are classified depends on the type of neurotransmitter that is released into a synapse. Acetylcholine is released by cholinergic neurons and is quickly removed from the synapse by the enzyme acetylcholine esterase. Neurons that release norepinephrine into the synapse are classified as adrenergic. Norepinephrine is slowly inactivated by monoamine oxidase. All preganglionic neurons in both the sympathetic and parasympathetic divisions are cholinergic neurons and secrete acetylcholine. In addition, all postganglionic neurons in the parasympathetic divisions are cholinergic. In the sympathetic division, cholinergic postganglionic neurons stimulate sweat glands. Acetylcholine binds with specific receptors on the postsynaptic cell membrane. Nicotinic receptors, when activated, depolarize or excite a cell. On the other hand, Muscarinic receptors, when activated, may excite a cell through depolarization, but can also hyperpolarize or inhibit a cell. Norepinephrine stimulates the postganglionic neurons of the sympathetic division connected to most effector cells. Depending on the effector, the adrenergic receptors on the postsynaptic cell membrane will be activated by norepinephrine to excite or inhibit the cell activity. Let's take a look at the hypothalamus. The hypothalamus coordinates the activity of the sympathetic and parasympathetic divisions of the ANS to ensure an efficient and balanced response. The sympathetic division dominates during a medical emergency, during exercise, when you are embarrassed, or when you react emotionally to situations because of the type of stimuli. The sympathetic responses are known as the fight or flight or alarm reactions. Sympathetic responses include pupil dilation, increased heart rate and force of contraction and blood pressure, bronchodilation of airways in the lung, vasoconstriction of vessels supplying blood to urinary, digestive, and reproductive organs, vasodilation of vessels supplying skeletal muscles, cardiac muscle, and liver, increased blood glucose from liver and fatty acids, and glycerol from adipose cells. In general, sympathetic responses provide a rapid mechanism that increases nutrients and oxygen to support increased metabolism and activity in organs needed to resist stress. The effects of sympathetic stimulation last longer and are more widely spread than parasympathetic effects. And there are three reasons for this. Motor pathways diverge to more effectors. The activation of norepinephrine is slower than the activation of acetylcholine, and norepinephrine secreted from the adrenal medulla during times of stress increases and spreads the overall effect of norepinephrine that is released as a neurotransmitter. Now let's take a look at the parasympathetic. When a person is relaxed, well-fed, or sleepy, the parasympathetic division dominates. Because of this, the parasympathetic response is known as the rest and digest reaction. Parasympathetic responses which oppose the support of physical activity decrease heart rate and force of contraction and blood pressure, bronchoconstriction of airways in the lungs, vasodilation of vessels supplying blood to urinary, digestive, and reproductive organs, vasoconstriction of vessels supplying skeletal muscles, cardiac muscles, and liver. In addition, a group of parasympathetic responses known as SLUD or SLUD include S. Salivation is increased. L. Lacrimation is increased. U. Urination is increased. D. Digestion and defecation are increased. In general, parasympathetic responses provide a rapid mechanism to promote the maintenance of normalcy, decrease energy consumption, and increase energy conservation.